school and high school, and is discussed in most college-level biology courses. We'll start by reviewing photosynthesis and the first part of this reaction, the light-dependent reaction. Then we'll discuss wavelengths that create usable light for photosynthesis and cover factors that influence the rate of photosynthesis in plants. We'll conclude by discussing some specific plant adaptations to different light environments. Photosynthesis is the process used by plants and other organisms to convert light energy into chemical energy. This chemical energy is stored in carbohydrate molecules like sugar, such as glucose. Photosynthesis is largely responsible for maintaining the oxygen content of the Earth's atmosphere and supplies energy necessary for all life on Earth. From this balanced reaction on the slide, not only can you see that photosynthesis changes sunlight into chemical energy, but it also splits water to liberate oxygen and fixes atmospheric carbon dioxide. In plants, long-term energy storage in the form of these sugars is produced by a subsequent sequence of light-independent reactions called the Calvin cycle. Photosynthesis is a two-step process. In the light-dependent reactions, one molecule of the pigment chlorophyll absorbs one photon and loses one electron. This electron is the first of many in the flow of electrons down an electron transport chain that leads to the ultimate reduction of NADP to NADPH. In addition, this creates a proton gradient, also called an energy gradient, across the chloroplast membrane, which is used by ATP synthase in the synthesis of ATP. In the light-independent reactions, also called the dark reaction or Calvin cycle, the enzyme rubisco captures carbon dioxide from the atmosphere and then uses the newly formed NADPH and releases 3 carbon sugars, which are later combined to form sucrose and starch. Not all wavelengths of light can support photosynthesis. The light reactions of photosynthesis require that pigments intercept specific wavelengths of light referred to as photosynthetically active radiation, or PAR. PAR designates the spectral range of solar radiation from 400 to 700 nanometers, also known as the range of visible light on the electromagnetic spectrum. This is the part of the spectrum that photosynthetic organisms are able to use in the process of photosynthesis. The meter pictured on the slide, the Light Scout quantum meter, is designed to measure the light that drives photosynthesis, or the PAR light. This is also referred to as quantum light because it is measured in units of moles striking an area over time. Plant biologists quantify PAR using the number of photons in the 400 to 700 nanometer range received by a surface for a specified amount of time using the photosynthetic photon flux density, ppfd. ppfd is normally measured by moles per meter squared per second. Measuring quantum light can tell you if your plants are getting sufficient amounts of usable light. The specific light meter on this slide measures PAR in micromoles per meter squared per second. Notice the low reading in an indoor setting compared to a shady outdoor environment and an environment exposed to full sun. PAR measurement is used in agriculture, forestry, and oceanography. One of the requirements for productive farmland is adequate PAR, so PAR is used to evaluate agricultural investment potential. The photosynthetic action spectrum depends on the type of accessory pigments present. For example, in green plants, the action spectrum resembles the absorption spectrum for chlorophylls and carotenoids, with absorption peaks in violet blue and red light. Photons at shorter wavelengths tend to be so energetic that they can be damaging to cells and tissues, but are mostly filtered out by the ozone layer in the stratosphere. Photons at longer wavelengths do not carry enough energy to allow photosynthesis to take place. Chlorophyll, the most abundant plant pigment, is most efficient in capturing red and blue light. In land plants, leaves absorb mostly red and blue light in the first layer of photosynthetic cells because of chlorophyll absorbance. Green light, however, penetrates deeper into the leaf interior and can drive photosynthesis more efficiently than red light. Because green and yellow wavelengths can transmit through chlorophyll and the entire leaf itself, they play a crucial role in growth beneath the plant canopy. Several factors can influence the rate of photosynthesis. Normally, the rate of photosynthesis increases as the amount of PAR increases, until it plateaus at the saturation point. Theoretically, you could keep increasing the light intensity, but the light reactions will max out, so the rate of photosynthesis will not increase. The rate of photosynthesis and most other metabolic processes increases as temperature increases, also plateauing and then decreasing as temperature becomes too high. Often, an increase in light intensity, or PAR, results in or is associated with an increase in temperature, so it's not always easy to tell if effects on photosynthesis are due to light intensity or temperature, or a combination of both. The intensity and spectral quality of light exhibits large temporal and spatial variation. For example, the daily photon flux available for plants grown in the deep shade of tropical forests is more than 100 times less than that available for plants exposed to direct sunlight. The spectral quality of light available for plants in the shade environment is often different from that of the full sunlight due to filtering by the above canopy. Plants are also frequently exposed to rapid and irregular changes in light intensity. I sort of answered this question on the last slide, but can you think of other factors that might affect photosynthesis apart from the three we mentioned? Take a minute to review all of these points before downloading and listening to the lab activity PowerPoint. In the following PowerPoint, I will go over an experiment using different plant species grown in a greenhouse under different light treatments and the effects these light treatments had on these plants.